We often focus on muscles, for power and bones for structure.
Right.
But what about those crucial connectors in between? Tendons.
The unsung heroes, really. Connecting muscle to bone, absolutely vital.
And they go through some really significant changes as we age. So today we're doing a deep dive into some cutting edge research.
Yeah, looking at what's happening right down at the molecular level.
Exactly. And for you listening, if you want that core understanding, but maybe without getting totally bogged down in the super technical stuff,
Then you're definitely in the right place. We've sifted through the latest findings.
yeah, we're bringing you the key insights. Our mission today is really to understand the role of this specific protein. It's called Cochlin.
Cochlin, right. In tendon aging and also in repair.
Our sources are scientific articles, research papers, so get ready for maybe some aha moments.
Because it turns out this one protein, Cochlin, it might just be a really key player in keeping us moving well as we get older.
Okay, so let's start with the basics maybe. What are tendons? What's their main job?
Sure. So picture them as these really strong but also flexible straps or cords.
Connecting muscle to bone.
Exactly. And their primary function is just transmitting the force your muscles generate. That's what lets you move, basically.
And the framework, the structure within these tendons,
Yeah.
that's the extracellular matrix, right? The ECM.
Spot on. Think of the ECM as this intricate scaffold. It's made up of, well, lots of things. Collagens are the big one, but also proteoglycans, other proteins.
And it's this matrix that gives tendons their strength, but also their elasticity.
Precisely. It lets them handle all the stress and strain of movement.
But here's the thing, as we age, this ECM, it can start to degrade.
Yeah, and that degradation, it really impacts how well the tendons can handle load, and how well they heal if they get injured.
And we've known for a while, haven't we, that the actual protein composition of the ECM changes.
Oh, yeah. These studies, proteomic analyses, they basically inventory all the proteins present. And they show clear differences between young and old tendons.
So what are some of those differences?
Well, younger tendons are typically rich in these molecules, called small leucine-rich proteoglycans, SLRPs for short.
SLRPs. Okay, like what?
Things like fibromogulin, myomekin, aspirin. These names might not mean much, but they're key structural players in young, healthy tissue.
Okay, so that's younger tendons. What about older ones?
That's where you see a shift. Older tendons tend to show more intermediate filament proteins.
It's like the building materials change over time.
Kind of. You could think of young tendons with those SLRPs as being like tightly woven, really flexible ropes, whereas older tendons with more intermediate filaments might be more like a bundle of fibers that are maybe strong but less adaptable, less elastic.
Right, which might explain why they're more prone to injury sometimes.
Exactly. And these molecular changes, they directly link to the biomechanical decline we see.
Like what specifically?
Well, aged tendons tend to be less stiff. Their ultimate load capacity, the maximum force they can take is lower. Their viscoelastic properties change too, meaning how they respond to stretch and recoil. All this makes them more susceptible to rupture.
And slower healing too, right. That's a big one.
Definitely. Slower healing is a hallmark of aging tendons.
So, okay, these protein shifts are happening in the ECM. But it's not just the matrix itself, is it? There are other changes.
No, it's broader than that. Cellular changes are happening too.
Like the tenocytes, the cells in the tendon.
Exactly. Tenocytes are the cells responsible for maintaining that ECM. They're like the construction crew.
Okay.
But with age, their numbers can decline and their function becomes less efficient. They don't maintain the ECM as well.
So the upkeep isn't as good.
Right. Which makes the tendon more vulnerable, especially under stress.
And what about this fragmentation idea? Proteoglycans breaking down.
Yeah, that's another piece. These larger proteoglycan molecules can break into smaller fragments, sometimes called neopeptides.
And these fragments aren't just inert debris.
No, they can actually be disruptive. They can mess with the matrix structure further and even trigger inflammatory responses.
Okay, so degrading matrix, less effective cells, disruptive fragments. Anything else?
One more big one, non-enzymatic cross-linking of collagen.
Okay, break that down. Collagen fibers are the main structural ropes, right?
Right. Think of them like strands. With age, these strands can start getting stuck together, cross-linked, in a kind of random, non-functional way.
Does that make the tendon stronger? More connections?
You'd think so. Maybe initially it increases stiffness, but over time it's actually detrimental.
How so?
It leads to brittleness. The tendon loses its elasticity, its ability to stretch and absorb shock safely.
Ah, so it becomes more like glass than rubber.
That's a decent analogy, yeah.
That is
Strong in one sense, but prone to shattering, or in this case, microdamage and tears.
Okay, that gives us a good picture of the general landscape of tendon aging. Let's zoom in now on Cochlin.
Right, Cochlin. This is interesting because it was first really known for its role in the inner ear. Quite different.
Yeah, involved in hearing and balance, I think.
Exactly. But recent research, really quite new, has put it center stage as a critical regulator of tendon health, too.
And how did researchers figure this out? There was a key study.
Yeah, a paper in Matrix Biology Plus. They used a mouse model, specifically, mice genetically engineered so they couldn't produce Cochlin at all, knockout mice.
So they could see what happens when Cochlin is missing from the start.
Precisely. And the findings were quite striking.
What'd they see?
Well, even by 3 months old, which is pretty young for a mouse, like early adulthood, these Cochlin-deficient mice showed changes in their collagen fibrils.
What kind of changes?
The diameter of the fibrils was larger than normal. This suggests Cochlin is somehow involved in controlling how these fundamental building blocks are assembled correctly during development.
So problems right from the early stages. Did it get worse as they aged?
It did. By 6 months, the tendons without Cochlin were significantly less stiff and had a lower peak load capacity. They broke more easily.
6 months in a mouse is maybe
Like 8.
early middle age for us.
Roughly, yeah. And importantly, these mechanical deficits were even worse by 9 months.
So progressively weaker tendons without Cochlin.
Exactly. It really highlights that Cochlin isn't just for development. It seems crucial for maintaining tendon resilience over the long term.
Did they look deeper, like at the proteins and genes inside these tendons?
Oh yes, they did comprehensive proteomic and gene expression analyses. Basically a deep dive into what proteins were present and what genes were active.
And what did that show?
In the Cochlin-deficient tendons, they found a downregulation, meaning less activity or production of proteins involved in some really fundamental cellular processes.
Like what?
Things related to RNA metabolism, how cells process genetic instructions. Also, the production of other ECM components was reduced.
So the cells weren't building the matrix properly either.
Seems that way. And even proteins related to the cytoskeleton. The cell's internal scaffolding were affected.
Wow. So missing Cochlin has really broad ripple effects inside the tendon cells.
It really does. It suggests Cochlin is involved in coordinating a lot more than just, say, direct structural support. It influences how the cells themselves function.
Okay, so weaker tendons, altered structure, messed up cell processes. But what about healing? Did they look at injury repair?
They did.
Yeah.
And this is where things got, well, surprising.
Oh.
How so, despite all these underlying weaknesses,
Yeah.
the reduced stiffness, lower strength, when they injured the tendons of these Cochlin-deficient mice,
Yeah.
the tendons healed at a rate pretty much comparable to the normal mice.
Really? That is unexpected. So Cochlin is vital for everyday structure and strength, but maybe not so critical for the actual process of healing after a tear.
That seems to be the implication, yes. It suggests Cochlin's primary role might be in maintenance and long-term integrity rather than acute repair mechanisms.
Huh. So the big takeaway from this Cochlin research is...
The Cochlin is a really central player in keeping tendons healthy and functional over time.
Mm-hmm.
And crucially, it suggests that the natural decline in Cochlin expression that might happen as we age could be a key factor driving that slow, subtle deterioration of tendon quality, maybe even before we notice any obvious pain or injury.
That's fascinating. And it definitely opens up avenues for thinking about treatments, right?
Absolutely. If declining Cochlin is part of the problem, maybe boosting it could be part of the solution.
How might we do that? Like gene therapy?
That's one potential avenue, yes. Gene therapy to encourage tendon cells to produce more Cochlin, or perhaps even delivering the Cochlin protein directly to the tendon.
Has that kind of thing been tried?
There's been some promising preclinical work, meaning in lab settings or animal models, for similar approaches in other tissues. But it's early days for tendons.
Okay, what else? If Cochlin affects ECM production, maybe target the ECM breakdown.
That's another angle. Modulating the enzymes that break down the matrix, like MMPs, matrix metalloproteinases, and their inhibitors, trying to tip the balance back towards maintenance rather than degradation.
Could you combine that with something like exercise?
Potentially, yeah. Exercise is known to stimulate tenocytes, those maintenance cells. So maybe a combination approach, biochemical intervention plus targeted exercise, could be effective.
What about, like, physical supports, scaffolds?
Bioengineered scaffolds, yes. Designing materials that could be implanted to reinforce a damaged tendon. And you could potentially load these scaffolds with Cochlin or other helpful ECM proteins to encourage better integration and regeneration.
Biomaterials is a fast-moving field.
It really is. Lots of innovation happening there.
And we shouldn't forget the basics, right? Nutrition, lifestyle.
Definitely not. Supporting collagen synthesis through diet is important. Things like vitamin C, certain peptides rich in glycine, minerals like zinc, they all play a role.
And sensible exercise, like physiotherapy-guided movements.
Crucial. Appropriately dosed mechanical loading is probably one of the best things we can do right now to help maintain tendon health as we age. It stimulates those tenocytes.
Of course, all these potential new therapies, gene therapy, MMP inhibitors, scaffolds, they need testing, right?
Oh, absolutely. Rigorous clinical trials in humans are essential to prove safety and effectiveness before any of these become standard treatments. That's a long road.
Yeah. Is there potential for, like, early detection? Using Cochlin, maybe?
That's a really interesting thought, biomarkers.
Sure.
Could we, for example, measure levels of Cochlin fragments or those neopeptides from ECM breakdown, maybe in blood or urine?
To see who's at higher risk of tendon problems.
Exactly. Or to monitor if a treatment is actually working at the molecular level. Early identification and monitoring would be huge.
Okay, so wrapping this up, the big takeaway seems to be Cochlin is way more important for tendon health than we might have thought.
Absolutely. It's vital. Influences collagen structure, mechanical strength, even how the cells inside the tendon work.
It's one of those aha moments, understanding how one specific molecule can have such a big impact.
And for listening, thinking about your own body, how it stays resilient as the years go by, knowing about something like Cochlin gives you a sort of deeper layer of understanding.
Right, it's not just wear and tear, it's specific molecular processes changing.
Exactly.
So here's a final thought to leave you with. We found out Cochlin seems really key for long-term tendon health, for maintenance, but maybe less critical for that immediate healing after an injury.
Right, that surprising healing result.
So what might be the long-term impact if we mostly focus our efforts on treating tendon injuries after they happen, rather than finding ways to proactively support tendon health, maybe at that molecular level, before things go wrong?
Focusing on cure rather than prevention, essentially.
Yeah. Could understanding Cochlin and maybe other molecules like it shift how you think about proactive strategies for keeping your body moving well long into the future? Something to chew on.